Glad to welcome you here this afternoon. Thanks. I'm Ed Lazowski from Computer Science and Engineering, and it's a great pleasure today to introduce my friend David Shaw, who will give us a really interesting interdisciplinary talk. David was a Stanford PhD student where he was actually housemates with our own Alan Borning and then the best man at Alan's wedding, so they go back a long way. He was a faculty member in computer architecture at Columbia. Then he went off and built probably the world's first big-scale successful program trading house on Wall Street and did that for a number of years, and then probably 7 or 8 years ago left to form D.E. Shaw Research, where he has built the Anton supercomputer for biochemical simulation. This talk is co-sponsored by Computer Science and Engineering and Biochemistry. David is colleagues with David Baker in Biochemistry. So David, without further ado, thanks for joining us today.
Thank you, Ed. Now, let's see. Is this dark enough here that you can see for movies? Okay, great. So I thought I'd start by just telling you a little bit about D.E. Shaw Research, which is an independent private sector research lab. And the business plan is a long-term one. It's something where we're investing in fundamental scientific research because that's the only way to solve the problems that we're interested in. And the basic idea is to use computational methods to develop new drugs that hopefully are more effective and also safer. And the enabling technologies are ones that allow you to do biomolecular simulation of a particular sort, which is called molecular dynamics simulation. And probably some on the biology side know something about that, but I'm going to go through a little bit just to explain what the background is, and then I'll probably go through some of the computer stuff in an elementary way at first, just so I can equally bore both sides of the house. But the basic idea of this simulation from our viewpoint is to understand how proteins move, looking at the three-dimensional structure of proteins and then watching as they move. And the reason for that is that that's where a lot of interesting biological function takes place, is in the shape of the proteins and what parts are exposed at various times. I'll give you a sense of that in a few minutes. And then the goal is to use that information in order to design drugs, to find places on a protein where small organic molecules like drugs can bind, find new ones in ways that can bind very specifically to cause their effects, because proteins turn out to be where the vast majority of the action happens in the body that's targeted by drugs. So basically, you can think of it as a drug development company that is intrinsically tied to technological advances in order to make money and also in order to hopefully do something that'll save some lives at some point. So let me start just by giving a little basic biochemistry for the computer scientists here. The first part you'll all know about probably, which is that DNA, we've decoded the genome, which is a sequence of DNA, and we now know it's not the only thing, but one of the key things that DNA does is it codes for proteins in the body, and specifically every 3 base pairs. Let's see where this shows. There we go. Every 3 of these base pairs codes for 1 amino acid, and there are 20 different amino acids. Just think of them as beads that can be strung on a chain. And then the linear sequence of that chain determines the three-dimensional structure. So not only inside the organism, but in most cases inside just a little glass of water with some salt in it, you'll see that primary structure, that order of those beads, determining the three-dimensional structure of the eventual protein. And that goes, roughly speaking, in 2 different stages. The first is to fold up parts of the protein into different what are called secondary structures. So some of them look like helices, things that go around like that. Some of them are these strands that often go next to each other and form sheets. These are different elements of secondary structure, but the interesting part comes when those things form and then fold into a more complex 3-dimensional structure. It's referred to as the tertiary structure. And in some cases, what you see is lock and key interactions between proteins or between proteins and drug molecules. So that's a lot of what we're interested in. Now, where are we? Well, we've decoded the genome, but you can sort of think of that as a parts list for what goes on inside a human or an animal. But we don't actually know the structure of most of those proteins. We know a lot of them. But in fact, if we really want to understand the three-dimensional interactions, we want to know what all of those look like. And there's some categories that are particularly interesting where we know very few of them. And we also don't know, and this is sort of closer to the ultimate goal, how they work together, how these things fit together to form what amount to individual machines in different parts of the cell. So to understand a large part of the biology, and especially the pharmacology of different drugs and potential drugs, it would be really useful to know more about the three-dimensional part and also how things move. So that's what we're really after. Now, this is a simplistic, kind of view, which is from somebody who was trained as a computer scientist, which is, I see the world as, there are two basic approaches. The first one is black magic, which is laboratory experiments, where people go into laboratories and have complicated devices where they shoot energy at small things, get different patterns and interpret them. This is obviously impossible, but people manage to do it. But there are limitations. It's very hard to even get still pictures of what's going on, much less movies. There's some places where you can get that, but we're really interested in our group at understanding those motions. Now, the other one, which is more familiar territory for people with my background, is simulation, to basically start with the basic physics that affects the way the forces among the various atoms and how they can expect to move and form structures and then interact with each other, start with that and then simulate to see those things occur. And the gold standard for doing that at the level of things the size of a protein is something called molecular dynamics simulation. Now, that's not totally accurate. What you're using is physical approximations, and in fact, the phenomena that we'll be talking about are in fact quantum mechanical ones that are imperfectly modeled by these classical models. But you simply can't analyze things with any known technology at a quantum mechanical level of the size that we're interested in and over the periods of time that we're interested in. So this is about the best that people have been able to figure out how to do so far. And let me just walk you through the various steps. So the first thing you do in an MD simulation, molecular dynamics simulation, is you divide time up into a lot of very tiny time steps. And unfortunately, they have to be on the order of 1 femtosecond. And the reason for that is that what we're going to do is integrate Newton's laws of motion, and if you divide it up into time steps that are too long, that process becomes unstable, you get energies blowing up, the simulation becomes meaningless. So you have to simulate at a level that's sort of comparable to the fastest vibration of the systems, which happens to be the covalent bonds between bonded atoms. And that forces you to do this very, very often. And there's a lot to do in every step. So the first one is, there's a little bit of a lie, but think of it as you're calculating the forces among all pairs of atoms in the system. Now, you can actually cheat. With this atom right here, you don't have to account separately for the way it interacts with things that are really distant from it. But the key inner loop, the thing that takes the most time inside an MD simulation with ordinary hardware at least, is this quadratic problem of interacting this guy with all of the ones around that are within a certain interaction radius. So then for each one of those pairs, we then evaluate what's called a force field. It's a biophysical model of the forces. And this actually is in the form of potential energy. You have to take the gradient of that to get the forces, but I just want to show you the general form. I won't go through every term, but it's an additive function where the first three terms I'm describing here are often referred to as bonded interactions. And those are ones where you either have two atoms bound together and they have a characteristic length that they want to stay with. So you basically have a formula that approximates that as if it were a perfect spring. A little harmonic constraint there. It goes like that. Then the second one is when you have three atoms and it has a preferred angle. And this final one is for when you have four atoms and you look at how it's rotated this way. Now, I won't go into any more detail. This has to be done, but computationally it's not as big a deal because each atom is bound directly or within a small number of links only to a small number of other atoms. And that being the case, it doesn't really take that long, even when you have to calculate a little function like this. It's the part that is down below that takes most of the time. And the first part is something very familiar to all of you, I'm sure, which is it's simply Coulomb's law. If you have two particles that are oppositely charged, they tend to attract. If they're, I said oppositely. If they're the same charge, they tend to repel. And the strength of that interaction is, in energy terms, 1 over r, or in force terms, 1 over r squared. And that turns out to be the main thing that's going to take time, simply because the 1 over r doesn't decay terribly fast, and so you have to consider things that go out pretty fast. And to a lesser extent, there's something right here, which this 1 over r to the 6th term you can't completely ignore, but you don't have to account for it very far away. So once you're done with that, what you do is calculate all those forces with that function, move the atoms by a tiny bit, and then repeat it over and over again, and you're integrating Newton's laws of motions, and you see a complete trajectory of the molecule. I've sort of short-circuited a few things here. It's not quite right, but think of that as the computational task that we're solving. So let me just run you through a little Gedanken experiment, which is, what if it were possible to do these simulations with perfect accuracy and arbitrarily fast, literally? So you could run arbitrarily long simulations that perfectly modeled what went on physically. So why would that be interesting? Well, again, from a computer science viewpoint, the thing that would be interesting is that you can model arbitrary computational, you could do arbitrary computational experiments to model reality, and that gets you out of the world of having to do these messy wet lab experiments and into a world where what you've got is a process of data analysis, which is very, very hard to analyze large amounts of data, but it's kind of comforting to know that it's there and you can find it, and if you can analyze it, you can get the information you wanted. And once you did that, you could then, if you're interested in what structure proteins have, you could simply look at them form. Start with something where they're extended, and in most cases, we know that that forms into the three-dimensional structure. So we could always get the structure, and we could find out exactly how it did it. And we could watch the dynamics. Those trajectories are very interesting, both from a scientific viewpoint and also in terms of designing drugs. In many cases, that's critical to designing drugs. So these are very interesting things to want to do. And the only problem is that MD, first of all, isn't infinitely fast, and also isn't infinitely accurate, although I'm going to talk about that only toward the end of my talk. So let's just talk about speed. I'm going to be talking today about a special purpose machine that we designed over a period of years. And before the development of this machine, which is called Anton, the fastest that you could simulate sort of a typical size protein on the fastest supercomputers available would only get you a simulation of about 100 nanoseconds per day, a tenth of a microsecond. And the longest simulation ever done before this machine was 10 microseconds long. It's a heroic feat on a big supercomputer. Yielded some very interesting data, but that's still a fairly short amount of time in terms of a lot of the structural changes one might want to look at. And so there are many biological phenomena, not all of them. Some take place over days or weeks, but there are an awful lot of things that are interesting biologically and pharmacologically that occur within this window of around 10 microseconds to a millisecond. And that includes things like major structural changes, and I'll show you a movie that sort of illustrates that in a second, interactions of proteins with other proteins and with nucleic acids like DNA and RNA, and most importantly to us with drug molecules or candidate drug molecules that we might want to use to intervene with the structure of proteins and shut down things that cause disease processes or maybe even cause them to work again. And there's another one that I'm going to actually talk about for a little time, and I'll explain why in a second, which is protein folding, the process by which this long string folds up into a three-dimensional structure, which is interesting to us for three different reasons. I'll get to that in a second. So first, just in terms of what Anton is, it's a special purpose machine for molecular dynamics simulations. And I want to emphasize that because of largely what it isn't. It's not somehow, a brilliant solution to the problem of having very fast general-purpose supercomputers. We're certainly not smart enough to do that, and there's some intrinsic reasons why it's a very hard problem. Because what you really want to do with that kind of a machine is to simulate things that have nothing to do with biology, a wide range of applications. But we had the luxury of trying to build a machine that would just do one thing very, very fast. And fortunately, it was an application where we could take advantage of parallelism, of something where you used a huge amount of arithmetic all in parallel using custom designed chips that were designed just for this application. And the result is something that turns out to be dramatically faster for those kinds of simulations, but much less flexible. So even though in theory you could do arbitrary computations with this machine, first of all, it would be really difficult to program, but it also wouldn't be any good. It would either be a lot slower or it's just not the right kind of solution. We think of it as a laboratory instrument, sort of the equivalent of a computational microscope, and that's all. There are 13 of the machines that we've now built now. One of them I'll mention in a few seconds, which we made available to the outside research community, the not-for-profit institutions, mostly universities. The other ones we're using ourselves. And each one of those is capable of millisecond scale simulations, which is much longer than was possible before. And just from a computer science viewpoint, let me just, actually for some of the biologists who know about work that's going on elsewhere, which is absolutely first quality research, but it's very different. The problem we're solving is to run single, very long simulations, which is quite different from running a very large number of short simulations. Also interesting, but you get different things out of it, or running a very short simulation of a very large molecule. And the thing that's unfortunate about it is that what we're trying to do is much harder. If you're going to run either a very large number of short simulations or short simulations of long ones, in either case, though for different reasons, it's much easier to parallelize, to break up the problem into a lot of little pieces and then execute them on a cluster full of processors that don't have to communicate that much. And the reason that's important is because when you scale up, one of the typical things that kills you, it typically will ultimately be the thing that kills you, is the communication among the processors. Not for all problems, but for many of them. So we've got a harder problem, but the machine is designed from the ground up. Both the hardware and the architecture were designed together to minimize the amount of communication and also to use the data once it got to where it's going in a very efficient way. So let me give you some bottom line numbers. This is kind of a random assortment. I just tried to pick out some kind of ballpark numbers. It depends how you measure them. But single processor codes, what you would do on an x86-like processor, running the code that tends to be fastest for those kinds of runs, get about 2 nanoseconds per day. And on the fastest parallel supercomputers, the numbers about 2 orders of magnitude larger than that, depending how you measure it. This is on, I should just say, on a particular fairly standard benchmark called Dihydrofolate Reductase, which has about 25,000 atoms, including all of the water molecules. We explicitly represent all of the water molecules that we're simulating. That's not always done in some applications, and often you don't need to, and there are certain advantages to not, in fact. But on our machine, that's what we always do, and that's the standard benchmark task. So get a couple orders of magnitude here. On Anton, we're able to get about 17.4 microseconds a day. So it's a large speed up, another couple orders of magnitude. And what that allows us to do is run much longer simulations that were, than were possible before. Again, just a couple of illustrative examples of some of the longest simulations that were done. On a single processor, we have, on the order of a couple microseconds. On a high-performance cluster, this was Klaus Schulten's work, which was the previous record. It was about 10 microseconds of simulation time on a different protein. These are slightly different proteins. For the same protein, Anton 1, it's one of the early simulations we did, got about 100 times longer simulation, and we were able to see some big motions and actually repeated protein folding and unfolding. And I'll show you a little bit of data from it. In that run. And then this is a more recent one. I just changed the slide. We now have about a 2.1 nanosecond simulation of the N-terminal fragment of the L9 domain. It's a ribosomal protein. We'll probably run that a little bit further, but we're getting about to the point where we have the data that we need. So we can run millisecond-long simulations fairly routinely. Most of what we do is shorter, more like 100 microseconds or even a little bit less, but they're much bigger numbers. And soon I'm going to show you one of those simulations, but, oh, right, I just wanted to mention something. I mentioned that we've made a machine available for researchers. And at this point, what's happened is the national academies have allocated time to 45 different research groups who are using Anton. It's a machine that's located in NRBSC, the National Resource for Biomedical Supercomputing, and it's available just to not-for-profit institutions. NRBSC receives funding from the NIH, and we're hoping they'll get a renewal of funding. It's a complicated issue, so that we can continue to do this. But one thing, there's been sort of a misunderstanding and some suspicion that we heard about sort of secondhand among members of the academic community that somehow we were somehow siphoning off government grant money to make a profit. And we are, in fact, a for-profit company, but this particular transaction is not designed for that. NRBSC gets the money from NIH, and we actually receive some reimbursement for certain expenses at cost. But the goal for doing this from a business viewpoint is, in the long run, anything that turns MD into something that plays an indispensable role in drug development for various reasons we think is very good for us, from a commercial viewpoint. But it's not the sort of thing where we're trying to somehow extract federal money to help ourselves here. It's more long-winded than you probably need, but because that's circulating around, I wanted to address that. This is Anton, and it's very pretty and has blue lights. And now I want to move on to an MD simulation. This particular one isn't very interesting biologically, and in fact, it was the first relatively long simulation we did, and it was largely to check the hardware and make sure that the MD calculation was stable, which it was. But I'm going to show it to you just because I've got more detail on this slide, where each one of these little spheres is an atom. And I wanted to show you how they move. And this is something I didn't see at first. I had read a fair amount over the 10 years I've been doing this about what proteins did from fragmentary experimental work, what they would look like over this period of time. But until I saw the movie, I didn't quite get it. None of us really got the picture of what happened until we actually saw a movie of it. And the other thing to explain is that because the fast motions of these proteins occur on a much faster timescale. This is, roughly a quarter of a millisecond. And as I mentioned to you before, each individual frame of this MD simulation is like a femtosecond. Millisecond is 10 to the 12th of those individual frames. If we showed everything here in the movie that was happening, you'd just see a big blur because the vibration would wipe it out. So this is kind of a time-averaged, smooth or low-pass filtered view. So let me show you what that looks like. What you'll see is a smooth version of some of those vibrations and some rotations of certain angles here. But the interesting part is that you'll see long excursions from what's called the native state, the state that it just sits in biologically if it's not doing anything in particular. It's not static. Every once in a while, it will sort of stretch out and then come back to roughly the same place. It's actually not, in this case, exactly the native state. So you can just get a feeling for bigger motions than you were able to see before. And in fact, at the end of this, I'll just show you, I'll just be eyeballing it, but what the longest simulation, the Schulten simulation from before, the record simulation would look like on this sort of a time scale. So it'll be about this, about that long. So it's not that it's uninteresting, and in fact, if you unsmooth it, you see very interesting things going on here, but you're not exploring the kinds of big structural changes that for many biological processes really explain what's going on. So that's what we're trying to do, is see those big changes. And I'm going to show you one other. This was the first millisecond simulation ever done, and it turns out it's also the same molecule, bovine pancreatic trypsin inhibitor, that Martin Karplus many years ago did for the first MD simulation ever performed. And all I want to really show you here is some of the structural changes you just don't see. This is a short segment, but we had to watch a long time. These are water molecules, and the colors just identify different ones, but this is where it's interesting. This big structural change, which hadn't been really suspected, caused water to flow through this. We don't think that's biologically meaningful. We don't think it's functional, but it's the kind of thing you would miss if you were doing the kind of simulations people did before. And the sorts of things we do with that is to then take a look at clusters of closely related conformations, we call them. It just means a three-dimensional structure. So each one of these dots is, it's sampled, but each one of the dots is one three-dimensional conformation, which is a 3n dimensional quantity, but they're just, it's projected onto 2 dimensions so you can see the clusters. And then the colored ones separate out different clusters. If you do a clustering algorithm, you get several different distinct clusters with relatively infrequent transitions between them. And then sometimes we look at the transitions over a period of time. So here you see the same colors and different y-axis now. What that's measuring is the distance in some sense. It's the root mean squared distance between the native structure that it sits in an experimentally determined crystallographic image and where it is right now. So this is the one that happens to be closest to the native structure, and it's going to then bounce up to something that's more different. This is actually 2 different colors, but that's just a coincidence because they're very different structures, but they happen to be about the same distance away in this metric from the native structure. And we see transitions here. And although I'm not going to really show you it, the kinds of things we're interested in is what kinds of motions occur on this fast time scale where you're doing local explorations, what kinds occur when you make these infrequent transitions. And we found this very clear separation of time scales. So the big changes occur on the order of 1,000 or more times more slowly. And they're very distinct. The kinds of parts of the protein that move are very different. It's sort of the outside of the protein, these side chains that rotate look like a liquid, and then you have a big structural change. And if you look at autocorrelations, you can measure some parts of it and see what's moving. And then the liquidy part, turns out it doesn't repack in a very systematic way. It forgets where it is. I know this is impressionistic, but it just gives you a feeling for the kind of questions we were trying to answer here. But so that's what we do often with these kind of simulations. And although we weren't particularly interested in the case of BPTI and function, we're often interested in mechanism, in trying to impute mechanism from what we see how the protein works as a machine, and then hopefully understanding enough about functionally how it works that we can try and figure out how to intervene here when something is broken. And I'm actually going to show you a whole set of applications, each very quickly, that relate to specific diseases and specific drugs that have been designed by other people at this point, and just give you a feeling of how that interacts with these slower changes. So first let me say something about protein folding. It actually is involved in various disease processes like Alzheimer's disease and Parkinson's, cystic fibrosis. And these are diseases that involve misfolding, where instead of folding into the correct structure, what they can do is, move into a different structure long enough to aggregate together and form a big clump. And even though in Alzheimer's disease, we actually don't know exactly what's going on, but we know this is characteristic. And I could go through the others, but there are diseases related to the folding process and to misfolding. But it's also just a fundamentally important biological process, and understanding certain things, the original reason we did it, about folding, we could use that as sort of a test case to improve the accuracy. First of all, test the accuracy, and then improve the accuracy of the force field, of the biophysical models that drive our simulation. And so let me just start by, this is not a terribly long simulation. This is on the order of, I think this one's 10 microseconds or so, maybe even 5. This is a very simple protein fragment called the villain headpiece, actually a double mutant version of that was designed by Bill Eaton specifically to fold fast. And I'm just showing it to give you a feeling for folding with a different representation. So this is that secondary structure representation where we have, this happens to be just a helical protein with 3 distinct helices, and the green is the native structure it's trying to find. We just started with a random polypeptide chain, and then you'll see it based on physical forces alone, finding the native structure. So these physical forces, they tend to form these helices early, and then ultimately these are kind of greasy residues, phenylalanines, that want to be all together, just like oil in a pot of spaghetti, and then this guy forms last. The only reason I'm showing it to you is so you can visualize what folding is. This is not, there was nothing new about this. Other people had folded this particular protein, even with a much simpler continuous model of water. But what we were able to do is take a look at some proteins that were kind of more interesting to us in various ways, and then take a look at the folding and unfolding of those proteins. First of all, doing it through the whole process and doing it multiple times so we could examine what the pathway is, what the sequence of changes were by which a protein, this particular small protein, it's a WW domain, how it folds. And so here you just see the folding process occurring repeatedly. This is the folded state right down here, again, RMSD, so this is close to what you, very close to what you observe. There's always a little vibration around the native state. When it goes up here, that's unfolded, where it kind of goes into a more or less random configuration. It's not really random, actually, but something that isn't folded. And that goes back and forth a number of times. So one of the things we looked at was what the sequence was. And I won't go through this in detail, but this basically maps out certain different parts of different distances within the molecule so that you can trace what's folding. And here's a little cartoon of it that's easier to see. There are a couple structures here. There are 2 what are called beta hairpins, hairpin 1 and hairpin 2. What we see is hairpin 1, we observe that in simulation every time forming first. It may accidentally go along hairpin 2 for a tiny bit, but you always see a stable structure in hairpin 1, then hairpin 2, and there's some other stuff that goes on. But for now, just think about that one order, and that's reproducible. Unfolding is the opposite of folding. Everything happens in the opposite order. So here's an example of a computational experiment we did. We were interested in understanding why hairpin 1 folds before hairpin 2 in the process of folding the overall protein, the WW domain. And so what we did is snipped off the 2 hairpins and folded them in isolation. First of all, we found they did in fact fold into something that looked like the native structure in isolation. And hairpin 1, which is the one that we see folding first in a folded protein, actually forms a little bit more slowly, not dramatically so, but more slowly than hairpin 2, but it's more thermodynamically stable. And for those of you who don't deal with this stuff all the time, just think of it as more stable. Once it gets there, it tends to stay in the structure much more than hairpin 2 does. So it is just a little snapshot of a few things to look at, but I just wanted to give you a feeling for folding, and then I'll point back to that in a couple of cases. Another test that we did, essentially experimentally. We don't do the experiments, we collaborate. We don't have anything wet in our lab at all. But what we did is used our MD simulation results and what we learned from them to design a new protein, which was a beta protein, beta sheet protein, and we designed it to be something that we thought would fold faster than any previously known beta protein. So we made, it was actually 3 mutations in this same one that we had simulated, and then Martin Grubla in his lab actually made the protein, watched it fold, and indeed it folds about 3 times, a little more than 3 times faster than the previous record. This isn't any sort of definitive evidence. The way it really works, and works in general in science, is you build a model and then you don't do a test that makes sure that says it really is true. What you do is you come up with potentially refutable hypotheses, throw tests at it, and see whether it breaks. And if so, you're in trouble with your theory. If not, all it means is so far it's held up, but you keep looking for more tests. So it survived this particular one anyway. Now, this is one that I was allowed to show for the first time the last couple times I gave the talk, but I'm not supposed to say anything about it yet because it hasn't been published, and there's 2 people who did most of the work want to be able to have it in print before they give the details. But basically, we wanted to see whether we could fold some other small proteins. And these are some of the proteins, and what we found is that 11 of the 12 ones that we tried, and they're taken from a systematic set that Bill Eaton actually, again, enumerated in a much earlier paper, 11 of the 12 folded. There was one more that wasn't thermodynamically stable at that temperature, so we made a change in that. We actually simulated a different variant that was, and that one finally folded. This does not mean that we can simulate the folding of all proteins. These are really small proteins. No question in my mind that we cannot, in fact, computationally fold all proteins. First of all, it would take too long for large proteins, but in addition, it's very unlikely that the force fields that we and the rest of the community are using now will be adequate to do that, and it would take much too long again. But it's kind of interesting. We learned some things from it, which we'll hopefully be able to say something about pretty soon. And let's see, one other thing I want to get to, then I'm going to shift into computer science a little bit. So here, what we're doing is taking a look at drug binding to proteins. And I have to say this part was not my idea at all. And in fact, had I been asked, there was a fellow in our group, Yibing Shan, who, if he had asked me first and said, I have this idea, I want to try this, I would have said, Yibing, yeah, it's a great idea, but realistically, what are the odds it's going to work? We'll use up Anton time, and you're busy working on stuff that's higher priority. I wouldn't have said, no, you can't do it, but I would have discouraged it. And the only thing is that Yibing was right, and it actually worked. So what he did is pretty simple. He took a, yeah, you can see it here. He basically took a protein that is known to be involved in leukemia. It's called the Src kinase. And then a small organic molecule that's known to bind to it. And in fact, it was originally developed as a drug, although it has toxicity, so it's not used at this point. And then all he did is he put the protein in a box full of water, put one of these small molecules, put this PP1 small molecule in there with it, and waited to see if it would find the place on the protein where it attaches, because it was known from experimental evidence it did, and it was known where. This is something called the ATP Binding Pocket inside this kinase and a number of others, and it was known that PP1 bound in exactly this way. So this guy over here is that molecule that Yibing threw in the box, and you can just watch it, and it's not a complete result that is we know what to do with, but it's kind of exciting that it does this. So here's a drug, and it searches around here just based on the physics, and it actually pauses here, which is kind of interesting. In a metastable intermediate state, does that pretty reliably. And then it finds its way into this binding pocket. You don't see the waters, but it's displacing some waters in the same way as the actual native pose. And when it stops, it's really hit the experimentally observed version. So it's kind of, it was unexpected to me anyway that would work. I should say that this is a long way for something, the immediate temptation was I got all excited and said, my God, you could then try drugs, design one, put it in a box of water, not only find whether it binds, but where, and then there are measurements that you can actually do to figure out how tightly it binds in a thermodynamic sense. You could follow the kinetics. You could see where it kind of gets stuck like it did over here. There's a lot you could learn, and you can see the obvious mapping to drug development. Now, it's not that simple. This took quite a while. Generating good compounds, to try this out on, and you couldn't do this with 1 million compounds, is very, very hard. So at least hard for us. So it's not clear we're not there yet. It's not like this is a magic way to design drugs, and we don't know exactly where it'll fit in. But it's kind of exciting to see the thing find its binding site and then bind there. And we've now got a couple other examples. Oh, this is the same one, which is just showing the water molecules. And in fact, it's a very snug fit. This is when it finds its binding site, if you consider the water, it's fitting very neatly in there, which is what the drug really wants to do, among other things. So here's another one. This is also binding to sarkinase. It's a drug called dasatinib. And it is an actual drug that's approved for leukemia. And it's known, again, where that binds. And this is another experiment Yiping did where he just threw it into the box. I mean, it's harder than that, but put it in the box, and it fairly quickly, in this case, found the rough right area. It's going to pause here, and it's actually kind of interesting because, maybe it already did it. A little groove opened up here, and it got distracted by it for a second, but then eventually it goes to exactly where it's supposed to. And then this is a different kinase. It's the epidermal growth factor receptor, which is, it's actually a family of 4 different proteins that are implicated in several different indications, but in this case, for this drug, the drug is approved for the treatment of breast cancer. It's called lapatinib, and I guess it's hidden right now. This is the last one I'll show, but the drug's going to search around again, and it's going to find the right place. And again, there are a few false places that it is before it finds the right one. One very interesting thing about this is, this isn't like a new finding, but is what's called induced fit. In the process of having the drug molecule fit into a protein target, it's not just that the drug itself moves around, it's also that the protein moves around, making space for it. And the induced fit part is when the drug actually gets there, it can actually change the shape of the protein locally. And many methods to computationally identify the drug binding site and look at how well it actually works as a drug assume a rigid protein. They might actually start with several different snapshots of the way the protein could be, and then what's called dock all of these little drug molecules into it, see where they could fit, not through the actual dynamics, but just kind of moving it around like this in all the possible rotations. But there's some kinds of drug binding which probably occur in areas that aren't even pockets. They're not, usually these kind of drugs bind to little depressions, and they fit very nicely, and they have certain kinds of chemical interactions. But in some cases, we don't think they're there. This has been postulated. We're seeing some examples that I haven't shown you where it appears to create its own pocket where you couldn't have predicted where it was going to bind and then bind in there. But this is all very, very preliminary, intriguing from the viewpoint of drug design, but early stage. So switching to computer science for a sec, Anton was designed by a classic procedure of co-design. Very early in the project, when I came up with the overall architecture for the Anton machine, I wrote up a little white paper just for members of the group, and it included not just the architecture, but an algorithm that fit that architecture. And if I'd been trying to design a new architecture to run standard algorithms that have been used on individual processors or standard commodity clusters, that wouldn't have gotten the speed up nearly that we really wanted. And conversely, if we had just started with the assumption of standard hardware, I don't know if I said this backwards. Yes, I think I did. If we figured out a new architecture for the standard algorithm, that would be too slow. If we figured out new algorithms for standard hardware, I'm all scrambled there. But both directions, it would be too slow. What you really had to do is find an architecture and an algorithm that fit with each other. So the Anton architecture is based on that and a bunch of other stuff. Most of the hard work came much later. So what does it do fast? There are a number of things, but the real engine, the part that was the inner loop, which it does really fast, is particle-particle interactions. It's that quadratic loop of all possible pairs of atoms. And it's actually used in other places too. It's actually not just particle-particle, it's point-to-point interactions. So I won't describe what this is, but for some distant interactions, we have an algorithm which is a variant of other work. The important stuff came before us, where it has a continuum model of things that are far away, and it turns out that involves a grid, this technique. And so interacting particles with the grid is one of the key things you have to do, either to, in one part of the algorithm, to spread charge onto the grid, and in the other, to interpolate back from the grid to other particles. And Anton does that very well too, but they're not separate things. They all use the same kind of hardware. We also, I mentioned the architecture and the algorithms. The underlying biophysical models also really had to be, not exactly changed, but we had to look very carefully at what kinds of physical models we could do well in hardware, which kinds of things we couldn't, where we wanted flexibility, and where we really had to have really stupid hardware that was just really fast. And I'll show you a picture of that in a second. So the different models we support, they exploit the architectural opportunities that were available to us in flat silicon. How could we map it out? So one I mentioned is we explicitly represent water molecules. That's very useful in some cases biologically. Drugs often bind, for example, by displacing water in a pocket and then replacing the favorable interactions and doing other funny things with entropy and so forth. But the other reason for it is it just would have, if we didn't have that because of the architecture, one part of the simulation would run really fast. That would be the surrounding water, which is actually most of what you're simulating. Most of the atoms are in the surrounding solvent environment. And then the part that was the protein itself would take longer, and all the other processors would sit there doing nothing. So there was sort of no point from an efficiency viewpoint either. So we thought about a lot of those interactions. Also thinking about future, more accurate models, there's something called polarizability, which I probably won't have a chance to describe in any detail, but it's basically a more accurate way of representing the electron cloud that surrounds all these nuclei in the protein than the way that's typically used in most force fields now. And so our machine, again, uses these particle-particle interactions with these sort of imaginary particles, imaginary charges, to model polarizability, where the electron cloud is dragged and you have an induced positive and negative charge. It's just one example, but the idea was look at the biophysics, find a way to map that onto the architecture, and vice versa. So I'll go over this really quickly. There are two areas where Anton gets its speed up. One is huge amount of hardware, great arithmetic density for very specific kinds of computational, very specific types of computation, and then using flexibility and programmability only in a few places where A, it's really necessary, and B, it doesn't happen so often that you have to waste a lot of area on it and put in a lot of stuff, a lot of memory use, going out to cache and so forth. In fact, just one little systems design thing of interest is the algorithm, to its first approximation, runs with no access to external memory. All the random access memory it needs is on this custom chip itself. For big systems, it actually streams some things in, and it's a little bit less efficient, but that's part of what we wanted to avoid, because you didn't have to fill up the chip with cache memory. On an ordinary x86 processor, on a single core processor, the vast majority, all but a few percent, is actually things other than the arithmetic. And with the Anton chip, the vast majority is actually doing useful computation for MD, not for other things. And then the other is carefully choreographed communication between the chips and within the chip. So going through very quickly, there are 32 of these pipelines on the chip This is where that inner loop of particle-particle stuff goes on, and all you really need to know here is every one of these blue things is doing some arithmetic, so there's a huge amount of parallelism. And the other thing to know is this is really not very smart. The data, just particle position data flows down, forces or potential energy come out the other side, and it never loops up, which causes all sorts of problems. It has a looked up table. There are a few functions that can be specified, but really, really dumb, but very, very fast for this application. And then this is the method I came up with. Just the key thing is for those interactions, it's asymptotically better than the ones that were used before for interacting the particles. It's what's known as the range-limited version of the classical n-body problem of physics. But the important thing is the constant factors are such that it tremendously reduces the amount of communication among processors. There's a whole history. I should give credit to other people here. Mark Sneer proved some things about lower and upper bounds, and in fact, you could figure out from that that there was a constructive proof that you could build something with these asymptotic properties, but it wouldn't work for this, and another proof that shows it's the best you can do. But just to show you the practical part, I won't even explain what this is, but blue is bad. Basically, we divide up space into little boxes, and each box contains all the atoms in one piece of space. And the problem is it has to interact them with all these. So here's the amount of volume from which it has to interact, bring in these atoms by communication. And the typical way is bring in that atom from somewhere else to a box where at least you have one of the atoms sitting. Intuitively, that seems like it has to be better. What I proved here was that that's actually not, and that the superior way to do it was to interact them on neutral territory in a box that was separate from both of them. And it's a little bit complicated how it works, but you can prove it does all possible interactions. And so in this case, you don't see a dramatic difference, but there's less volume here that gets imported, and hence less communication. It doesn't matter with a small system. This is actually almost a wash. But if you go to 512 processors and then 4K, and this is kind of the effective range where we operate, the traditional method, the amount of data that you have to transfer roughly turns into a hemisphere with, at a finite range, slightly flat tops. And this one turns into an it imports from an infinitesimally thin, what I call a plate, and an infinitesimally, narrow tower. So as it gets bigger, there's less communication. There's also some latency stuff that other group members came up with that becomes very important when you move into this regime. That's about all I'll say. Let me just quickly dash through 2 minutes worth of other pharmaceutically relevant studies. One that is involved in kidney disease, and that's a study of antiporting, which is where you send one ion one direction through a cell membrane, another ion coming in. This one, you send a sodium ion out and two protons go in. And we were able to find 3 particular amino acids that controlled this process. It was a model that was introduced like 50 years ago, but we could actually show how it worked at an atomic level of detail. This is a potassium channel. These are channels where it actually still moves ions, but in just one direction and very selectively. This is like a hole. It's called a pore. And these are potassium ions. It won't pass other kinds of ions. And so we could watch the process of conduction and some other stuff. We could sort of build a model of why that closed. So this is the flow of these things. And we tried to take a look at what closed it and came up with a model for why, with the different potential different voltage across the membrane, there was a set of changes that resulted in what we call hydrophobic collapse that closed the channel so nothing could go through. And there's a movie of that I won't show. This is G protein coupled receptors. This is one particular one called the beta-2 adrenergic receptor, which is implicated in asthma and some other things. Did I say the other one? This has a long list, but neuropathic pain is actually one of the most important. There's some kinds of pain from nerve damage that you can't treat. In diabetes, it's a big, big problem. So that's an important target there. GPCRs are the drug targets for the majority, by some counts. I've seen between 30 and 50% of all drugs interact with this class of drugs. And I won't have time to show you, but there was a confusing thing. There was only, at this point where we did the research, there were only 2 known structures of this very large class of compounds, and there's biochemical evidence that they should have done something. It's formation of something called the ionic lock, and you should have seen it in both, but you didn't experimentally. So in simulation, we found you actually do see what you would expect in simulation, but not much of the time, and we could show why that was. This one's involved in leukemia. It's work that we did with John Currian at Berkeley. This is a very successful drug. It's sort of the poster child for a highly targeted drug that's actually keeping people alive for a long time, but not all. 10% don't respond, and then a few people, we now know as of last week that, or 2 weeks ago, that actually it's very good. If you survive initially, you're probably not going to relapse, and it used to kill pretty much everybody. It's a drug called Gleevec, and so we wanted to understand the mechanism at an atomic level of detail of a previously known change, structural change, and then we looked at how Gleevec blocked that, and we made some predictions based on that to test our theory about the how changing the pH ought to change the binding affinity, how well you get Gleevec binding, and John ran experiments which matched that very closely. Then we simulated various mutations, made predictions about that. John again tested them, and that worked out, quite accurately. So we have a reasonable amount of confidence in that model. And I think this is the last one. Presbyopia, you usually call that farsightedness, but when you get to my age, usually it's between age 40 and 50, you'll probably know this, that you start needing reading glasses, and it's because the lens of your eye no longer accommodates. When you look at something close up, it's supposed to change shape, and then when you focus on something far away, it changes back. Now, there's a protein in the eye, the most abundant protein in the eye lens, called aquaporin 0. The aquaporins generally are water channels, but this one, for some reason, doesn't pass much water. And by doing some studies and calculating in simulation some pressure differentials here, we were able to come up with a hypothesis and evidence, but I wouldn't call this conclusive, that phenomenon, that pressure and the way it distributed, might be holding these protein fibers, lens fibers, together in a way that preserves the structural properties and preserves the refractive index of the lens under conditions of accommodation. So I think that's basically it. The only last slide I wanted to give you was speed isn't everything. Accuracy is very important also. And we know now that very long simulations are useful for some kinds of applications, but clearly they're not accurate enough for all applications. If we could do infinitely fast simulations with infinite accuracy, we could, it's a little oversimplified, but we could design drugs and save huge numbers of lives very quickly. We can't. There's a lot more research to be done, and a lot of it is on what is inaccurate about the force field models that everybody is using. And there are a lot of suspicious things that people have as candidates, but we don't know which ones are the real problems. So the big question is, can we improve the accuracy of the force fields we're using? And as it turns out, very long simulations give you a new way to do that. You can run experiments that hopefully can turn up some of those problems. And then the question is, how efficiently? Do you need a different architecture? These are all open questions. We're very encouraged. We believe it'll play a significant role in drug design, but there's a lot open. And this is sort of like finding a silver lining, but even if we get negative results, we as a field, even if we find out that our current force fields aren't accurate enough to model a lot of things that we'd want to, the hope would be that this gives us some insights that could be biologically relevant and also pharmacologically relevant. So hopefully we understand how to go to the next step and learn something about the physiology that's important, both in terms of normal behavior and pathophysiologically. So that's all I've got. Should I?